and welcome everyone to today's talk presentation as part of the Enjoy Science Seminar Series. This is a series of educational opportunity that is organized by the Office of the Chief Scientific Officer here at MD Anderson, Dr. Giulio Draetta, with the intent to share exciting presentation during this time of very limited in-person, at the bench, and in the clinic activities. So I'm very excited about the presentation of today, and I would like to send a word of encouragement here from the MD Anderson Cancer Center, and I would like to thank you, the Office of the CSO, for the opportunity to learn and share the great research and translational research and clinical research that is conducted at our center with many friends around the world. I will now give the microphone to my colleague and friend, Dr. Kalin, that hosts this series, and he will be introducing today's speaker.
Thank you, Alessandra. Good morning to all the large family of MD Andersonian. Good morning to the world, and it's my pleasure to introduce a dear colleague and collaborator, Professor Han Liang. This webinar is named Enjoy Science, and I think Han, it's a wonderful example of a young and bright scientist who truly enjoys science, and he's making really impactful discoveries for patients and for the science. He's a bioinformatician, the type of scientist that everybody likes because they are doing wonderful things on the computer, but also he has a wet lab in which he's testing his hypotheses from bioinformatics to experiments. Han is a professor in the Department of Bioinformatics and Computational Biology. He grew in rank wonderfully quick. He's a star. He grew from 2009 to 2019 from assistant professor to professor, exactly because he's doing extraordinary impactful research, and it's easy if you check online. He's publishing paper after paper in great journals. He is one of the essential people from the Cancer Genome Atlas working group, from ICGC working group, and many other big data working groups. And in fact, his talk today, it's about how artificial intelligence is helping us in order to understand big cancer data. And with this said, thank you very much, Han, and let's run the show. Good luck, and thank you all for being here with us.
Great. So now, hello, could you, okay, now I show my slide. Do you see my slide?
We can see them.
Okay, great. So thank you, Alex and George, for a very nice introduction and the invitation. It's really my great pleasure to give a talk. And also, especially during this special time, I hope everyone are staying well and healthy. So today, I'm going to talk about how AI help us address the challenge of big cancer data. So this is different from my usual research talk, where we introduce a background, define a problem, and present a large-scale data analysis. So today, I really want to talk about how this AI can make fascinating stuff, how this AI can potentially transfer our research. So before I give the result of my presentation, I want to ask a few questions. So first one, which of the following is most different from the others? A PhD in bioinformatics, B, in computational biology, C, in biostats, D, in theoretical biology, E, a large pepperoni pizza. The answer is D, because all the other four can fit a family of four. Of course, this is just a joke. Basically, I believe everyone, as long as they enjoy their job, work hard, can always make a decent life. But let me say, when you contact somebody for a bioinformatics analysis, how long can you wait for the result? Say 1 year, maybe too long, right? 1 month, it's okay for some people, very patient people. 1 week becomes reasonable, right? How about 1 hour? And then even 1 minute. So I guess the correct answer is E. By the way, that's the why you need to stay at it until the end of my talk. So last question. So in this era of big biomedical data, what you would expect AI can do for you? Say A, generate some meaningful hypothesis, so you can further test in your lab. B, recognize some meaningful pattern of complicated data. Hopefully that's pattern, but meaningful pattern, and then you cannot recognize by your eye, right? C, build this accurate model for classification, prediction. D, do a time analysis of your interest. Discuss the result like an expert. Even write a paper and grant with you together. How about all of them? So that's why I want to show you a solution today. So this is, of course, just a teaser, but I want to give you some hints about where I'm going. So sometimes when you do this big data mining, just like you face this big mountain. So there are so much data, you don't really know where to look for. So you have to look deeply until you see there could be some pattern. Is there something familiar? Maybe yes, maybe not. How about I give this contest? So actually this indicates that the DNA sequencing cost was reduced dramatically in the last 20 years. So actually the speed even beat the Moore's law, which is for computer CPU storage cost decrease. So I also want to use this as one example that nowadays for our cancer researcher, we have this too much data. Sometimes we just don't know how to dig in, how to get a meaningful result, unless we have very effective tools. So today actually it's a very exciting time for cancer research. Let us just review some typical process for cancer discovery. Usually we start from this patient cohort, we found something unusual, maybe some variation could be driver gene expression. Hopefully from there, we want to understand whether there's an impact on cancer development. We try to understand some mechanism. So we do some manipulation in cell line, often knock out or overexpression that gene, see the effect on cell proliferation, on cell invasiveness. And then hopefully after that, we want to understand, get more idea. We want to design some therapeutic opportunity. We want to identify some drug and potentially treat that, kill the cancer, or hopefully that marker as a biomarker. And then if everything looks good, we do this, validate everything in vivo, usually through mouse model, look at the efficiency of drug efficiency, and then look at phenotype mechanism. And then if everything good, we move to clinical trial. So I should say this whole process actually is very time consuming. If you start from this very beginning to the end, it even take you 5 years, is a fast track, right? So, but nowadays we have this unique opportunity to take advantage of this big data to accelerate each step of this process. So for example, we from the TCGA on ICGC, where you have this comprehensive molecular data for a large patient cohort. From there, you can easily identify some high frequency variation. And then from there, hopefully you have some hint we want to focus on. Then we also have this large scale cancer cell line collection, very detailed molecular profile for each cell line. And also recently from Broad and Sanger, they also have this cancer dependency map where they use CRISPR or SHR knocked out each gene to see what's the phenotypic effect. And then after that, that will directly give insight into the mechanism. And then after that, we have this lot of drug pharmaceutical resource such as GDSC on connecting map, or even cancer target discovery development program. So they also generate a lot of drug, large scale for screening and also for a lot of detailed perturbed drug profile. So using this data, you can very quickly generate hypothesis, take much faster time to move to next stage validation and experimental testing. So, but on the other side, this big data also present great challenge for cancer researchers. So when we talk about the big data, from the beginning, this data volume is big issue. Now we not talk about megabit data, we not talk terabyte data, even petabyte data. So this huge amount of data, and then also there's a lot of different data type from DNA, RNA mutation, metabolic, even epigenetic, single cell. So there are so much different data type, and then each data type need a different tool, different visualization. To mention on the top of that, you want to integrate the data set across data type, get the result, and then you want to test the robustness of your observation in one data set across different cohort and so on and so forth. So this is also present a huge challenge for cancer researchers. Just to give you one example, the TCGA data, many of you familiar, this actually takes 10 years for large population of cancer researcher. Finally, we get a major cancer type, comprehensive molecular data for 33 cancer type, each with large patient cohort, usually 500, even 1,000 sample. And then you have this multi-dimensional characterization from mutation, gene expression, demethylation, even to RPPA, and also a lot of clinical data. So this is probably the most digested data set for the community. But even for this data set, it's still, you have so much new discovery nowadays, every day. So over the last 10 years, my group actually developed a lot of user-friendly tool for help people analyze this TCGA data. So from the early days, we developed this software identify survival marker, and then more the cancer proteomics atlas. So this really focus on MD Anderson unique RPPA platform. And then TERIC, this is a popular software for studying long non-coding RNA in cancer. And then also MCLP, this is for studying cell line, selecting cell line model system. And then more recently, we have this FACEMIC annotation, the functional effect of somatic mutation in cancer. And then just last month, we studied the mitochondria through the SAGC dataset. So this basically, each tool here hopefully provide a solution to each individual dataset on specific problem. For example, for this the cancer proteomics atlas, you just see this very nice interface. After that, you just click a few buttons. You can do some very common analysis. For example, you want to see if these two protein markers, they are highly correlated on a protein, they really show some differentiation between different breast cancer subtype. Protein expression shows survival correlation in cancer type. And also you can see this global pattern through cluster heat map on network. So especially we have this very fancy next generation cluster heat map developed by my chair, John West and Brad Broom. This really effective tool help you see this global pattern. This is just one example. And then in the last years, we have collectively, we have more than 80,000 people to use all these tools across more than 100 countries. So our goal is really try to make those tools as easy, very easy to use and user-friendly. But I know no matter how hard I try, I always below what you need. There's partially because there are just too many big data, right? What I just talked about is just TCGA. Then how about ICGC? Now we have 25,000 data. How about this biobank from the UK? Now we soon will get 500,000 people's genome data. And then this all of us, we have more than 1 million of the data. And then to mention this kind of ENCODE, epigenetics, GTAX. So there's so much data, essentially this huge amount of data just make people very challenged to people to digest so much data simultaneously, and hopefully from there get some very clear clue where to go. Second thing is, firstly, the tool what I developed is really defined for specific data set, for specific predefined analysis, where you just only can do the job where they want, the predefined in the software. The last challenge is there's just simply so many tools. Sometimes you just don't know which tool you should use. And then even a tool try to be user-friendly, there's still a learning curve. Sometimes you finally understand how it works, they update. And then later, sometimes you don't use the tool for a certain while and you forget it, right? So that's a learning curve, especially for some people have no computational background. That's really a challenge. So that's why the key question is, how user-friendly is the user-friendly? I think people can always say it's easy to say, but difficult to do. So how about we make the computational analysis? It's easy as when you say that, it's so easy to do, just like you can say that. For that's why we get the idea we want to introduce this kind of AI intelligence. So this is, take my group several years, we basically developed this software we can call that analytic platform called Dr. Anchorite. That's really a natural language oriented platform, can help you analyze and visualize all kinds of big cancer genomic data. So basically you can directly talk to this kind of software by natural language. So instead of you define the data set, identify a question, you can just raise question through a chatting interface. So here I can show you some example. Basically, now you can see this is the landing page, our tool, Dr. Anchorite. Okay, and then we develop all kinds of analysis, but basically for today's purpose, we can quickly show some demo tool. Basically, this is more like a chatting, like what we use, like a WhatsApp on Facebook, on WeChat. So essentially, you can just ask your question. For example, you can ask a question, say, just directly type whatever question you meant. For example, you just want to first get a simple summary about the data set. So how many cancer types in TCGA? So the Dr. Wright will understand your question, and then they began to confirm. After you confirm that, they began to do calculation. They generate this result. This bar show the progress, and then once they finish, they immediately show this result to you. And then here basically show this data summary, not only cancer type, but also the sample size in this bar. Or you can ask some more meaningful questions, say, you just want to ask, is there TP53 gene expression with gene overall survival? So for this question, you can simply type in breast cancer, and then the Dr. Wright essentially understand question. They ask which data resource you have. Then you can choose, say, TCGA. Then they confirm your data set, confirm your question, then show the result. So you can basically this you can see this progression, and then they show this result in the Kaplan-Meier curve. From there, you can further analysis. And then important, you can look at this figure, see low expression have better survival. And then even you can download this data as a figure directly in your slides on your publication. Important thing you see, we have this feedback to give this good or bad, to give us the feedback. So sometimes you may not really ask a question perfectly, right? You can ask a question, maybe you not complete at the beginning. So you just ask a question, do you want to do a correlation between P10 versus say this 2G? But essentially, this question is not very clear when you first raise this, right? So, but Dr. Wright, essentially, they can ask you, okay, then which data type you are looking for? Then you say, okay, gene expression. Then they confirm your question and do this analysis where where you basically just a few seconds, you see this differential gene analysis. And then you can see here basically show this 2 gene correlation in this box plot. And then you also can check different data set, the sample size, experimental correlation, include the p-value, FDR. And also you can even try different way, but data set, data visualization include the log transformation, and so on and so forth. So this, maybe sometimes you can ask a more complicated question. So you want to study, you just want to not only do 2 gene across the whole cohort, you just want to maybe study an important between 2 sub-cohorts. Say you want to know for one specific gene, is there differentiation between gender? Say because for the cancer we have kidney cancer, we have male and female. And then for this, by asking this question, again, they basically can do this kind of, Dr. Wright Uncle Wright can understand your question and then generate this result in seconds. You can see they really calculate everything in real time. That basically here they show this 2-group comparison in box plot, and then also you can check this block interactive, so you can see the detailed value for each box value. Again, you can save this figure for analysis. And then importance after that, you probably need to give us a rate, say cool, bad. So this feedback is important. Okay, then you may think, okay, this seems only can do something higher level data analysis. Could you pro some very dirty, long rate, difficult problem, say from the raw rates? That's also, for example, you want to do a differential analysis. But in this case, where you don't really have this, in this case, you want to really want to the doctor do a RNA-seq data. But in this case, you don't have normally you have the raw rates only. So the doctor rate, basically we will ask a few more questions about this data set. Say where suppose you have a FASTQ set assess number, and then they want to know which the data come from versus the tissue, and then you may even name this product a specific name because later help you with its result. Then you first step you want to just do RNA-seq quantification, and then in this case because it involves some hyper after a few moments you can take a coffee break. After a few moments you get a result. In this case, they generate this RNA-seq pipeline for the RNA rate mapping and quantification. On the right side, left side, they basically show all kinds of QC parameters about this pipeline, and then you have the feature count, and then basically you also can see the QC, whether this rate QC is good, do you have too many duplicate rates, or what's the general QC histograph, and looks this everything fine. Then you can see the quality score, baseline alignment. So, and then finally you can see the GC content. If this all QC parameter looks fine, they basically show you this, finally they can show this result, include the data, the facts. So here still checking some basic, you can either look at browse this quick QC report, and then finally this is your data set where you have the gene, you have the gene position, the length, and you have the normalized data set. And from this data set, you basically also have this exon here, basically TPM is the normalized gene expression for the gene. And then you have this raw data for you to download. On this data actually can further help this Dr. Uncle Wright to do further analysis. So Dr. Wright can also do this kind of complicated data using very different pipelines. So we have multiple pipelines. So essentially what we do is we have this but really you want to know where you this come from, right? So let me just tell you the behind after the analytical flow for this Dr. Uncle Wright. So essentially when you type a sentence, we use a natural language processing to parsing that into meaningful keyword, and then given this term, and then we build this Bayesian-driven probability model to help Dr. Uncle Wright understand what you are asking, which biological question you are asking. And then from there, we are mapping to the module, the analytical module. From the module, we understand what's the specifics, and then you identify the scripts on the software package behind this analysis. From there, we do finally a check, see whether we have sufficient information to do the job. And then given this job, we can determine whether this is a very simple job, so we can do this locally. But sometimes even you do a high throughput computing, probably for that job, we need to coordinate a high computing cluster, or this can be do in the cloud computing. And then we submit job. That job scheduler will coordinate whatever computing resource do the analysis. And from there, after they finish, they return all the results, and then they feed back to the Dr. Uncle Wright. Then Dr. Uncle Wright can talk to you, communicate this result directly. Okay, so but this is just the complexity for do this, but you don't need to understand this. The only thing is, from now on, you just have this tool. You can directly answer any question, just like you talk to a friend or talk to your bioinformatician colleague. So we also make a lot of effort, make this process very standardized. So we build this, each step is module. So because you have a lot of bioinformatics tool, we can have this tool first get a ramper. So this tool is not affected by our local environment. And then we can plug this kind of module, build this library. And from this library, you can all do this, all kinds of fancy analysis, from mutation analysis, complex heat map, network pathway, single cell analysis, even pan-cancer analysis, and the routine all statistical analysis. So this is, we believe this kind of tool also could have large impact into our research, not only make things straightforward and simple, but also could make our science more reproducible. You know, nowadays, reproducibility is a big concern about in the biomedical community. So on one side, a lot of papers, include those published in top journal paper, they're just not reproducible. On the other side, it's really take a lot of time and effort to reproduce some published result. Frankly, I understand this really very big headache, because number one, you don't really want to spend a lot of time to repeat something you already know. Even you confirm that, you don't get much credit. Second one, even for some analysis, some computational analysis, is still, if you do everything, it could be take a lot of time, right? So, for example, here is a paper published in Nature. Basically, this paper is a typical cancer genomic paper. They just describe the breast cancer in 600 breast cancer whole genome sequence data. And then in this paper, you see they generate this kind of typical figure, say the mutation, generate this map, right? The data set heat map, and even make a specific discovery for link RNA. But this, for qualified bioinformatician, they could reproduce this result by, say, probably need several days. But now, if the data is available, we can easily through this talk to Dr. Uncle Wright. On the right side, you see, just ask by talk to the question, immediately you see this result. Boom, boom, boom. You exactly can repeat this result. So if you take one step further, once we really set up this kind of natural language processing way for reproducibility, for each paper we publish, there's always a method material section. At least for the computational analysis, presumably that Dr. Uncle Wright can take the text literature in the method section, easily reproduce the computational analysis, because presume the data set, you make that every parameter detailed such a way that Dr. Wright can routinely repeat this analysis. That potentially can provide a fascinating way to check the reproducibility. Okay, so the next, this is, of course, just the beginning of the Dr. Uncle Wright. So what we really want to do, next step is try to get more data. So because currently we probably only get TCGA data, but eventually you can think we can get this large SCGC data, CCLE data, ENCODE data, GTEx data, and all kind of consortium data. The thing is, we can get NCI now have the movement to try to make those data more standardized. So as a user, you can easily we can easily load those data into the Anchorite. So you don't really need to worry about the local storage, the volume issue. You just talk to the Dr. Anchorite to answer those questions within data. And you also can upload your own data, basically talk to Dr. Wright, basically specify the location for your own data set, then they can do the analysis. On the second, we also want to add more pipeline. So currently we have just make some very common pipeline, include those mutation calling for whole exome, RNA quantification, ChIP-seq data, and so on and so forth. But this is only 20, 30 pipeline commonly used in the field. But potentially there are many more pipelines, there are more complicated analysis. So we can make all those analyses more pipeline, so you can choose different tools when you do the analysis. And then sometimes for the single task, let's say RNA-seq, you have multiple ways, maybe 10 different ways to call differentiation. You have different package. We can make all those multiple ways available, and then you can easily choose which way you want to call differentiation. Sometimes you may have, okay, give me 2 options, too many options, I don't know which way I choose. Then you can also ask them, based on other people data, which one is more accurate, or which one is more popular given the data usage. So all those questions, you can, Dr. Wright can give you some suggestion, which tool, which pathway, which pipeline you probably should use. Second part, we not only can give you the result, we can further interpret this result into the context of known literature. Because there's probably 3 million papers, right? After you see the pattern, the result we show, for qualified reviewer, usually they want to do some literature check, but this can be automatically done through, we can integrate this large amount of literature data into the backyard Anchorite database. Basically, when you see a pattern, say TP53 mutation with survival, they can immediately tell you whether this pattern already reported in certain paper, or this pattern even contradicts with already reported pattern. So in this way, you can imagine Dr. Anchorite now can discuss the result with you in the context of literature, can also search those literature in a more comprehensive way from keyword, and then get some imaging figure to support the argument. Further evolution, we can get more social interaction. I think nowadays more people actually spend a lot of time in online chatting. At least my training spend a lot of time on Facebook, WhatsApp, on WeChat. So you can imagine we can essentially make this Dr. Anchorite through this social network. From there, you can directly talk to Dr. Wright by voice. Now we have this feature. Basically, you talk to this, they understand the feature. Even you can talk to this even your multiple language, not only English. And then you can also have this security, basically define your own space where you contain some private data, where you Dr. Wright do the analysis only based on the public data and your private data. The private data only belong to you, so they can do the analysis for you. And also you can set up this kind of group chatting. This actually help you invite a working group, say you have small group discuss one product. Dr. Wright could be one partner. Basically, whenever you can imagine him is very smart, very knowledgeable, work really hard to answer any data-driven question you have. And then we want to make this more through computer into the smartphone. So basically, you can do this kind of intellectual Q&A answer question with Dr. Anchorite anywhere, anytime. So transparency. So I don't want to give you the impression that You just want answer question, we throw some answer. That's more like a black box. Eventually, we want to this process transparent. So when you have do this dialogue, you finally get a major report. From there, you know the version, the time, and then what's the detailed dialogue, and also you have this markdown language driven data report. From there, you know which data set we use, which pipeline we use, and then what's the parameters ruled in, and then where we get this result, what's the visualization tool. So everything becomes transparent. So essentially, after you have dialogue with this AI agent, what you can get is not only those graphs, also a detailed data report behind that data. So that report can bring to anyone to see whether that method is reasonable, whether the data can be result can be reproduced. And also make it open development. You can always think this Dr. Uncle Red more like a platform. So from here, we incorporate the data set. We have some, our team make some pipeline, but eventually they can also accept any pipeline contributed to other developer. So for example, in GitHub, a lot of pipeline, buying from my pipeline to already in GitHub, there we are going to make the API can automatically take this kind of pipeline into this into our platform. So from now you can easily call any public pipeline, not only from the one we implement, but also from public domain to do that analysis in our. And then they can support in multiple language from the MATLAB, Python, Perl, and JSON, and so on and so forth. Okay. So also want to mention very important thing. Remember, when we do this analysis, after each section, we want to give some feedback to the, it's up and down. That's also important learning process. So we can always use that feedback to improve our system. There are always some tasks, the sentence we cannot understand, we may get a wrong answer. There are always some tasks, we just don't have the ability to do that. But because this is a feedback loop, we can automatically take those feedback to make our nature NPL module more robust, make our those tools more transparent, more capable, okay? So you can imagine we start this product a few years, just like a baby. At this moment, you may see this can only do some routine analysis, some standard pipeline. But given the time, with the increased power of computing, we now eventually can make this doctorand very knowledgeable, basically know all the amazing data set, do all kinds of bioinformatics analysis, and can interpret the result in the context of knowing the literature, and then can challenge you using data-driven result. So this is really what we want to achieve. So by the way, this is my little daughter. She is only 3 years old. So finally, I want to just mention scientific research nowadays. I view it more like a co-investigation between human and AI agent, rather than something people are doing with AI-driven tool. So we are living in a fascinating time where you can imagine in 5 years, in 10 years, the way we are doing science will be very different from nowadays. So making this kind of AI-driven agent, essentially, we can make progress through this constant interaction between human thinking and AI-driven. So they both have their limitation and advantage. So human maybe have more subtle, maybe sometimes intuition, but agent have more computing, more systematic thinking. So when you combine the two sides, you probably can really resolve some fundamental challenge issues in the field. So here, just to share you the sentence and my favorite quote, the best way to predict the future is to create. Okay. So with that, I would like to thank my, so I have this wonderful group. So this really behind this is Dr. Jun Li. He was my former postdoc, now my junior faculty in my group. He really makes this the primary developer for this wonderful Dr. Ankorite. And then the core member also include Hu Chen, Yu Meng, and then Mei Ru, and also input from my other lab member. And also this cannot be possible. We really have this fascinating department level support. We have this very great software engineering team and the quantitative computing research group. So here is I want to thank for the all my funding support, and I would happy to stop here and take any question. Yeah.
Thank you very much, Han. This is a wonderful talk, and in fact, all of you see how the science will evolve in the next few years. It's really excellent, excellent development. So for now, Han, please check on the questions and from the chat, and please answer to the questions that you think can be answered now. And for all the attendees, if the time is going over, please contact Dr. Han Liang by email anytime. So Han, thank you again, and please answer to the chat questions. I saw already some questions are in, please.
Okay, so I try to, okay.
One question was about how are you dealing with the privacy of this information, because Dr. Ankorite will have all the rights to check here and there, about privacy of the data information.
Yes, that's actually what so accurate, the big data need to be validated. Yeah, so basically that's what I say. The next time we are now implement that, you not only get a result, you also will get a transparent data report where they basically tell you where we download the data, which language software we use, what's the parameter. So we use a markdown language, basically essentially can make that result very really make the result very robust and transparent. So that's right. Yeah, as I mentioned, in the future you want to this kind of thing transparent. And then there's a question about whether I think let me see. So this is, let me think, this also big data from for drugs, except for drug development. Yeah, so that basically that's it. Nowadays, because you have large patient cohort data, you have large cell line data, you also have large pharmaceutical data. When you integrate this together, I believe this is a very efficient way you can identify new therapeutic target, identify biomarker. The good thing is, personally, we want to generate data by ourselves and then to test the hypothesis. But today, there are so many generalized data already available. They can easily help you test that directly. And then there's also question, yeah, so think, yeah. Some questions say maybe there's an agent computational scientist. So basically, the AI agent essentially, yeah, I mean, essentially the software we developed. The thing is they can automatically take the feedback from the user and then using this feedback to systematic learning and improve its function.
Also, another question, Han, which is interesting. How do you know big data information are accurate? Because no, Dr. Wright will take all this data, who can be a mixed bag of accurate and non-accurate, and trying to give information. This information can be very helpful, but it's very dangerous for the people who don't know bioinformatics and don't know artificial intelligence, to rely on something which is not very well confirmed. Exactly what you were saying about data accuracy.
Yeah, so that's a very good question. So I guess we all worry about what we learned from the big data. I think there are two things. Number one, from our side, we try to make those data resource clear, and then we can load some standardized data. For example, in TCGA, we actually make another round effort to make data standardized. Actually, our department is the TCGA QC Genome Data Analysis Center, so we can load the best quality data as much as possible. Second one, you also can ask a lot of questions where you sort of know the answer to see whether the data set confirm some of the known knowledge. If, say, you already know certain thing is true, for that data set, you can constantly see this can confirm you already know the knowledge. Maybe that's a good sign that the data is good quality. And then when you generate some new hypothesis, you sort of that result you're more confident in the result. Second thing is big data, we often get multi-cohort data available. For example, you have TCGA, SCGC, and we are going to make a lot of other data available. You can always test whether certain finding is reproducible. So essentially, this is the same thing when you contact a bioinformatician for specific question. Bioinformatician also have to do this kind of analysis to ensure the QC is okay, the data is okay. And then you often try that to see whether that result in the other data set or not. And then if you see certain pattern consistent across multiple cancer, you sort of more reliable to use that. Yeah.
Thank you, Hans. So it's another question. Can you show an example from big data for successful drug development? So to focus a little what you presented on this concept of drug development that everybody now loves and everybody now is looking for.
Yeah, so that's what I will show the picture. Say, eventually, if you look at the whole process, it's like we usually start patient data. From that data, we want to identify some potential driver on at least some higher frequency marker, say EGFR mutation in lung cancer. But then the next question, you will see there's a high mutation in lung cancer. The next step, you probably want to see what's the mechanism. That's why you go to the cell line, you manipulate that, right? You do knock down that EGFR, or you overexpression EGFR, and you see they regulate that signaling pathway. After that, you want to see if we can find a drug potentially inhibit that, and then you do drug screening, or even sometimes you initiate. So this basically all this stuff, now on we basically can do this through data mining. Because personally, you probably only do one single gene-based cohort analysis. Now, from TCGA, you can routinely do this for all the analysis. And then once you identify gene, you go to that CCLE data, like a deep map, where you knock out each gene, you see the phenotypic effect. Immediately, you can confirm whether a high frequency variation is a driver event, really have some phenotypic effect. And then you can looking for some perturbation data like YO1000, where they knock out each gene and show the gene expression data. And then you can see which pathway they perturb. From there, you can know some insight. And then for GDSC or CTD2 program, where they generate a lot of drug screening data, you can basically see cell line with EGFR and without EGFR, which drug shows sensitivity. From there, you immediately get some drug candidate that potentially can trade this opportunity based on EGFR mutation. So that's what I'm saying is such kind of thing is nowadays, although I use EGFR is well-established example, but this kind of discovery can be now easily done through this systematic integrate multi-dimensional, multi-resource of the large cohort, large standardized big data set.
Another question, are artificial intelligence agents computational scientists?
So that's a very good question. So essentially, when you talk to them, you don't really hope one day we can reach a moment when you talk to them, you don't really know whether they are a real person or AI agent. You just told them like your bioinformatician colleague. You ask a question, they answer you in a very natural way. You even can make a joke. They understand what's your joke. So that's the hope. So, but right now, I guess if you talk to them, you still have to ask some scientific question. And then sometimes you think they will stuck there. So people know that's just an agent, not real people.
Can train OncoWrite in some jokes, you know? So one more question. When will be Dr. OncoWrite available, or how are you planning to to give it to the scientific community? What are
so this basically is already available. It's just drbiorat.org. So drbiorat.org. So you can easily Google Dr. Bio-Rat or Dr. Uncle Rat. You will see that link. Or if that's already in the public domain, so you can use that. Okay.
Thank you very much. Now there are other questions. If no, I want to thank very much to Dr. Han Liang. This really was a wonderful presentation to show the power of human mind and the power of artificial intelligence. And I want to thank very much to all of you who attended, because without your participation, such webinars are not possible.